Hello everyone. In this video, I'll be talking about biofilms in general and what is the role of biofilms in ENT. So if you see the definition of biofilm, it is an assemblage of microbial cells that is irreversibly associated. That is, it cannot be removed by gentle rinsing with a surface and enclosed in a matrix of primarily polysaccharide material. So it is basically a colony of microorganisms which are embedded in a matrix of a polysaccharide material like slime. Van Leeuwenhoek first observed the biofilms on tooth surfaces, and he is credited with the discovery of microbial biofilms. Now these biofilms they can form either on non-biological surfaces such as intravascular devices which are immersed in bloodstream, or they can also exist on mucosal surfaces within the body. So if we see related to ENT, then they can be present in tonsil or even the mucosa of the sinuses and middle ear. So they can be a cause of tonsillitis, sinusitis, or otitis media. Now, a biofilm forms in various stages. So first, there is the stage of reversible attachment of the planktonic or the free form of the organism to a surface. For example, if a biofilm is being formed by Staphylococcus aureus, so initially that bacteria, staph, will attach on the surface, and it is a reversible attachment. Then after some time, this attachment becomes irreversible and the bacterial community becomes covered in an extracellular matrix slime. So there is secretion of the extracellular matrix and the bacteria they get embedded and irreversibly attached in that matrix. Then within the matrix, the individual cells they communicate with each other via a process called quorum sensing. It allows the regulation of gene expression throughout the colony. So there is a communication of microorganisms within the matrix. And finally, when the biofilm becomes fully mature, it erupts and releases the free form or free organisms back into the environment. And thus this cycle keeps on perpetuating. So this is a pictorial depiction of the formation of biofilm. Here we can see that the bacteria or the cells they get attached irreversibly first to any surface. They multiply. They produce this extracellular matrix that is slime. It grows in size, and this is a fully mature biofilm with this is a matrix outside matrix and inside we can see that cells colony of cells. Now after becoming fully mature it erupts or ruptures, and these planktonic organisms or free form bacteria again they become free in the environment, again they can get attached on any other surface, and thus this cycle keeps on perpetuating. Now, examples of places in the body where biofilms are seen are dental plaque, then in ENT it is otitis media, chronic rhinosinusitis, even in tonsillitis, then colonization of lung in chronic respiratory diseases like cystic fibrosis, colonization of chronic wounds, osteomyelitis, intestinal microbiome, endocarditis, and some device-related infection. The implants that are used in the body, like knee implant or any hip implant, even they are known to get infected with biofilms. These are the common organisms which are known to form biofilms: coagulase negative Staphylococcus, Enterococcus species, Candida albicans, Klebsiella pneumonia, Pseudomonas aeruginosa, and Staphylococcus aureus. Now, what is the difference between a bacteria in when it is free in environment and when it is in a biofilm? For example, Staphylococcus aureus, if one it is free in the environment and other it is a part of a biofilm. So what is the difference that we'll see? So the first difference is that in a biofilm, the organisms they secrete an extracellular polymeric substance, matrix slime. But in free form, this matrix is not there. Secondly, in a biofilm, the microorganisms they have reduced growth rates. as compared to when they are in the free form in environment. And third, there is up and down regulation of specific genes. So as in the formation of the biofilm, we saw quorum sensing. It was a mechanism where there is up regulation of certain genes or down regulation of certain specific genes. So these are the differences between the two forms of the same bacteria. So what are the clinical implications of these biofilms? Now it has been studied that biofilms are very resistant to treatment. It is because of the following reasons. First is that presence of extracellular slime makes the penetration of antimicrobials very difficult. Then bacteria, they themselves exhibit reduced metabolic activity and they divide less frequently than as their free living forms, and this renders many antimicrobials ineffective. So we know that the antimicrobial agents, they mainly act on the division of the bacteria, but as in biofilms the division rate is decreased, hence these antimicrobials are rendered ineffective. However, the certain agents that act on ribosome, they have been shown to be effective against biofilms. Examples of such agents are rifampicin or macrolide agents. So, how can we manage biofilms? The first is that disruption of the biofilm by mechanical measures like debridement. So, if there is any slimy membrane over the tonsils or even in the ear or sinuses, it needs to be debrided. Then, if a device is involved with a biofilm, then if it is not being treated by medicine, then it may ultimately require removal. Then eradication of biofilm with topical agents such as mupirocin or manuka honey. These two agents have been shown to be effective against the biofilm. And then future research in this topic can be development of improved device surfaces that do not allow the attachment of the organisms to initiate the biofilm formation. So these are some of the strategies of management of a biofilm. Thank you.